Hello everyone. So today topic will be for the in continuation with the tissue culture part 2. Today I will be discussing about the types of culture. See, tissue culture is commonly used as a collective term to describe all kind of in vitro plant culture, but it should refer only to the culture of unorganized aggregates of the cell. So in culture, generally it is initiated from a sterile piece of a plant, and these pieces are called as explants. So what are explants? Explants are the like initiation of the culture from a sterile piece of a plant. Okay, and these pieces are called as explant, and it may consist of pieces of organs such as leaves, roots, or any specific cell type like pollen or a endosperm, right? So first type of culture we will be discussing is the callus culture. Callus पहले तो इसको समझते हैं. What is callus? Callus is an unorganized or undifferentiated mass of cells. ये कब बनता है? When the plant cells multiply in disorganized way. ठीक है? The process where tissues and cell cultured on an agar media forms callus is known as callus culture. So the process where tissues and cells, they are cultured on an agar media, and these tissues forms a callus. So this type of culture is called as a callus culture. So this callus can be initiated in a vitro by placing small pieces of whole plants or explants onto a growth supporting medium under a sterile conditions. What happens under the stimulus of endogenous growth regulators or growth regulating chemicals added to the media, the metabolism of the cells, which were actually in the quiescent state, is changed. Now they become active, and there is a beginning of the active division. So during this process, the cell differentiation, their specialization is going on, and they may occur in intact plant or they may reverse. So explant give rise to new tissues, which is composed of meristematic and unspecialized cell types. Got it? So I can write over here that the explants give rise to new tissues which is composed of meristematic and unspecialized cell types. So the callus formed using an original explant, they are called as primary callus. What are primary callus? Primary callus क्या है? जो callus form होता है original explant से, तो उसको हम बोलते हैं primary callus, ठीक है? और एक और होता है जिसको हम बोलते हैं secondary callus. Secondary callus क्या होता है? The cultures are initiated from the pieces of tissues which are dissected from the primary callus. इसका मतलब secondary कहाँ से आएगा? जो primary callus से लिया जाएगा. और primary क्या है? जो original explant से callus बनेगा. Okay. Now let us discuss about the pattern of growth and differentiation. Like a typical unorganized plant callus which is initiated from a new explant or a piece of a previously established culture has 3 stages of development. So we will be discussing about these 3 stages. The first is the induction of cell division. Second is a period of active cell division during which differentiated cells lose any specialized features they may have acquired and become de-differentiated. And third is a period when cell division slows down when or within the callus there is increasing cellular differentiation. So in many callus tissues, the relative level of the auxins and the cytokinins control the shoot and the root formation, right? Or an undifferentiated callus can be regenerated into whole plant by simply altering the concentration of the growth regulators, right? कैसे होगा? The roots are formed when auxin to cytokinin ratio is high. That means कि यहाँ पर क्या है, ऑक्सिन्स की कंसेंट्रेशन हाई होनी चाहिए एज कंपेयर टू दी साइटोकाइनिन्स. ठीक है, तब क्या बनेगा, रूट्स बन जाएंगे. एंड द शूट्स आर इनिशिएटेड when the cytokinin to auxin ratio is high. That means the cytokinin should be higher in amount as compared to the auxin. That will form the shoot. When the auxin is higher than the cytokinin, then the root, there is a formation of roots. अगर suppose intermediate level है, ठीक है, both the hormones, they will produce like completely disorganized callus growth. राइट अगर दोनों का मिक्स जो ऑक्सिन्स है और साइटोकाइनिन्स है, दे आर इन द इंटरमीडिएट लेवल, सो बोथ ऑफ दीज हार्मोन्स व्हाट विल दे डू, दे विल कंप्लीटली डिसऑर्गेनाइज द कैलस ग्रोथ. ठीक है, नेक्स्ट टाइप ऑफ कल्चर वी विल बी स्टडीइंग इज द प्रोटोप्लास्ट कल्चर. प्रोटोप्लास्ट पहले समझते हैं, प्रोटोप्लास्ट क्या है. प्रोटोप्लास्ट इज अ लिविंग पार्ट ऑफ द प्लांट सेल, राइट. एंड इसमें क्या है, इट कंसिस्ट ऑफ द साइटोप्लाज्म and the nucleus so protoplast is it's a living part of a plant cell consisting of the cytoplasm and the nucleus with the cell wall removed. जो अंदर का पार्ट है उसको हम बोलेंगे that will include the cytoplasm as well as the nucleus. So this is the protoplasm. Now this protoplast, it can be induced to reform a cell wall and divide if placed in a suitable nutrient medium. So what happens, a small culture of cell, eventually it will arise from each cell. Plant can be regenerated from such callus. At presently, isolated protoplasts are used chiefly in research when plant viruses are studied. I mean the plant virus infections are studied. Or it can be used for the modifying the genetic information of the cell by inserting a selected DNA fragments. Right. Another purpose of using this protoplast is they may be fused together to create a plant cell hybrids. So let us discuss about the methods of protoplast preparation. There are several methods by which the protoplast may be isolated. So I'm discussing about the methods of separating the protoplast. First is by mechanically cutting or breaking the cell wall. Second is by digesting away the cell wall with enzymes. So there is a use of enzymes to dissolve the cell wall for releasing the protoplast. See, enzymatic method, if one is using, It could be used as a one step method, जिसको हम बोलते हैं direct method. One step method. इसमें क्या है, प्रोटोप्लास्ट आर आइसोलेटेड डायरेक्टली फ्रॉम द टिश्यूज बाय यूजिंग 2 एंजाइम्स. और वो कौन से एंजाइम्स है? दीज आर सेलुलेज एंड पेक्टिनेज. ठीक है. One is a direct method, which is one step method. Here the enzymes are directly used, cellulase and pectinase. They are used for isolating the protoplasm. There is another method in enzymatic method, we can say, which is also called as two-step method, या फिर उसको हम बोल देते हैं sequential method. Sequential method, इसमें क्या करते हैं? The cells are first isolated from the callus or a tissue by using pectinase. So first they are using pectinase to isolate the cells from callus, and to this cell suspension of the cellulase is added. ठीक है. Then later we will use the cellulase, and we are using the cellulase to digest the cell wall and release the protoplast. This protoplast can then be collected. It can be purified using a centrifugation method, ठीक है, to separate the broken and the damaged cell from the intact protoplast. ठीक है, by taking the advantage of differing buoyant densities. And the last method is by the combination method of mechanical and the enzymatic separation. So third method is the combination of the above two methods. ठीक है, it's a combination of mechanical and enzymatic method or separation. So this is for this much. I am stopping over here. Next part we will be discussing about the cell suspension culture and organ culture. Thank you.